Я очень благодарен, да, за приглашение. Мне очень приятно выступить. И хочу рассказать про наши результаты. Должен сказать, что у меня очень мало опыта рассказа об этом по-русски, поэтому мне придется говорить по-английски. Но вопросы, конечно, в конце можно задать мне и по-русски, и по-английски. Надеюсь, что это нормально. Я думаю, вся литература на английском. Надеюсь, что проблем не будет. So I will switch to English now. I want to thank again the organizers for this opportunity to present our work on CD8 T cell dysfunctional states in cancer and infection that we studied using multiple different genomics data sets, including multiple different single cell approaches. So I want to start with a little background. Our work is concerned with cytotoxic CD8 positive T cells that are the central component of adaptive immunity. These are lymphocytes that work as highly specific and efficient serial killers, such as, for example, these 3 CD8 T cells surrounding and trying to kill a cancer cell in this image. CD8 T cells are essential in defense against viral and bacterial infections and in tumor immunity. And normally, for example, in acute infection, T cells recognize the antigen on an infected or a special so-called antigen-presenting cell. And with certain co-stimulation, these cells become activated, they expand, and then they differentiate to effector cells that kill the infection, and eventually to memory cells that enable more efficient response to the second encounter of the infection. But supposedly also the same response also happens in the tumor microenvironment, where CD8 T cells know how to tolerate normal cells of the body, but attack tumor cells that express new peptides or new antigens. However, in some circumstances, for example, in chronic infections or in tumors, as the tumor progresses from initiation to premalignancy stage to established tumor, or as the chronic infection gets established, some T cells do not mount a functional effective response and instead become so-called dysfunctional or exhausted. And in order to gain the molecular understanding, systematic molecular understanding of this concept of this state of CD8 T cell dysfunction, we decided to perform a systematic meta-analysis of genomic data for CD8 T cell dysfunction. We wanted to understand how similar T cell dysfunctional states are across different mouse models, particularly models of chronic infection, different tumor models. And it is actually very important, I think, to distinguish between model-specific effects, distinguish them from observations that hold more generally, because ultimately, of course, the goal is to generalize to human patients. We wanted to understand how T cells become dysfunctional and what underlies this differentiation process, and particularly what transcription factors are associated with this differentiation. And we wanted to understand at a much better resolution, at a single cell resolution, how are the chromatin states linked to heterogeneity of CD8 T cells. So we started with the systematic reanalysis of published bulk ATAC-seq data. As you probably know, ATAC-seq stands for assay for transposase accessible chromatin, followed by sequencing. It enables genome-wide profiling of chromatin accessibility landscape for inputs of very small number of cells, typically down to several thousand or several hundred cells, and also now available in a single cell variant that you will see in a few slides. In this assay, segments of DNA that are accessible to transposase are sequenced and then aligned to the genome. And peaks of this signal, we think, likely correspond to promoters and enhancers of gene expression. This assay has been transformative across fields, and particularly in immunology. And using ATAC-seq profiling in dysfunctional CD8 T cells in multiple different studies, it was shown both in mouse models of cancer and chronic infection that there exists so-called epigenetic imprinting of T cell dysfunction, which might be a barrier to functional rescue of T cells, for example, by checkpoint blockade. So we collected first the bulk ATAC-seq data sets for CD8 T cells from several recent publications. And here is an example on this genome browser snapshot of the data near one of the genes, PD1 or PDCD1, which is an important marker of T cell dysfunction and also a target of immunotherapies. Here, different colors correspond to different functional states as characterized in each individual study. And this includes profiles for naive cells, for functional effector cells, and also for memory cells in acute infection, and dysfunctional cells in tumors and chronic infection. And we did uniform preprocessing of all this data. We constructed an atlas of chromatin accessibility peaks of about 150,000 of those. And then also we used generalized linear model to remove batch effect, which enabled for us a direct comparison of the cell states across different studies. And after this batch effect correction, here I'm showing you PCA of all these data, where each point here is an ATAC-seq sample. It's a vector of ATAC-seq signal over the whole atlas of more than 100,000 peaks. And color corresponds to the functional state as it is profiled in each individual study. So the cell population was sorted, and a tax-seq was done at the bulk population, and this is what is reported with this color sorting strategy and the experimental details. So symbol corresponds to the source of the data here, and you can immediately see that, for example, all naive cells from multiple different studies all clustered together, all the functional cells, broadly speaking, also clustered together. But what's most remarkable is that dysfunctional cells from different studies, they all cluster very tightly together. And this includes T cells from chronic infections from multiple different studies, and also cells which are tumor infiltrating lymphocytes in hepatocarcinoma and melanoma models at different time points actually during the tumor development. And this includes endogenous and also adoptively transferred CAR T cells. And this includes also T cells with and without anti-PD-1 immunotherapy. So all these dysfunctional cells cluster together and separately from naive and functional cells. And this suggests to us that there exists a universal program of T cell dysfunction, which is very similar between chronic infection and cancer. And I think it's remarkable that this fundamental observation about T cells can be made by reanalysis of published data. And furthermore, as I mentioned already, some of these attack-seq profiles are also for important subsets of T cells. Indeed, it was recently demonstrated that dysfunctional T cells are heterogeneous, and while the majority of cells in the established tumor or chronic infection are in the state of terminal dysfunction, it was demonstrated first in mouse models and then actually in cancer patients, that there exists also a small subset of so-called progenitor or memory-like, stem cell-like or precursor dysfunctional cells. There are many names. They are characterized by multiple different surface markers such as CXCR5, SLAMF6, and also transcription factors such as TCF1. Very often these cells are simply called TCF1 positive. These cells are particularly interesting because they have proliferative potential and they can self-renew, and these cells respond to checkpoint immunotherapy. And in our analysis, we actually particularly focus on this subset of progenitor dysfunctional cells. And some of the attack-seq data in our compendium that we collected from previous studies was for this subset of cells. And of course, attack-seq measures the regulatory state of the cell, broadly speaking. And then the next crucial step for us in the analysis of attack-seq data was to understand the regulators, the drivers of these regulatory programs, transcription factors. So initially, we looked at the loci where these transcription factors are encoded. And here I'm showing you the results of analysis of differential accessibility, differential attack-seq signal, and differential expression in different comparisons. So the diamonds here, we call these plots diamond plots. The diamonds here correspond to multiple potential accessibility peaks associated with a gene. And little circles show the differential expression of a gene. So you see that this way we identify multiple different well-known genes, and including some transcription factors, which are consistently more highly accessible and more highly expressed in the progenitor cells as compared with the terminally dysfunctional cells in the chronic infection shown on the left, and also as tumor progresses on the right. And now this is where, so this was about where transcription factors are encoded as genes. But now using attack-seq data, we can also look at where transcription factors bind, maybe indirectly. For many transcription factors, we know that they bind to DNA in a sequence-specific manner. And this is also often represented as a motif. We found all significant matches of all transcription factor motifs in sequences of all peaks of chromatin accessibility atlas. The assumption here is that if a site is highly accessible and there is a motif for that transcription factor, it's a proxy for us of transcription factor potentially binding in this accessible site. Then to associate transcription factor binding with cell states, we use a negative binomial generalized linear model where we predicted absolute levels of accessibility in each cell state using this prior expectation about where transcription factors might bind in this atlas of chromatin accessibility peaks. And then using this, training this model, we inferred transcription coefficients corresponding to transcription factors, which are a proxy for us of transcription factor activity. And we summarize the inferred transcription factor activities. I mean, first we show, and I skipped that, we show that there is a consistency across different replicates and also even across different studies for what we observe in terms of these derived transcription factor activities. And then we summarize these inferred transcription factor activities over replicates on the same condition and across studies. And we this way, we identify many well-known transcription factors and actually even hypothesize about some new transcription factors potentially associated with different functional T cell states, including the progenitor state. That is our main focus. Now we did all this analysis at the bulk cell population level using publicly available data. But there is a lot of evidence, as I also showed you, that CD8 T cell responses are heterogeneous. So the next step for us was to look more closely at one of the models, particularly at the LCMV model, and to study this divergence between functional and dysfunctional responses. For that, we used single cell ataxic data that we generated with our collaborators using 10x platform. And we were particularly interested in identifying the progenitor subpopulation and studying it across different conditions. And we look very early on, relatively early on, after the initiation of the after T cell activation, because we want to look more closely at this divergence between functional and dysfunctional fates. And our previous analysis, as I showed in the previous slides, demonstrates that this divergence happens quite early. I think okay. So this is the summary of the data. If you work with single cell data, you probably have seen these kind of plots many times. We reduce dimensions and use UMAP for that, and this is what is shown on the left for the two samples. And also we use clustering, of course, to make sense of the data. And we use multiple different ways to functionalize the clusters in the data, including by comparing with our bulk ataxic atlas that we constructed. And we think that at least one of the clusters, among many things that we can observe from this data, we think that one of the clusters is very clearly corresponding to progenitor or this memory-like state of dysfunction. But what is very remarkable is that in this cluster, we also observe not only cells from the chronic infection, from the infection with clone 13 of LCMV, which results in the chronic infection, but also from the cells responding to Armstrong clone of LCMV, which is known to result in acute infection. And we think that these cells in this cluster, number 10, from the acute infection, actually are so-called memory precursor cells, which are also known to be progenitors of more differentiated cell states. And also by applying this, the same predictive model of transcription factor binding that I showed you previously for the bulk data, we think that we find this model very powerful. And we are able again to associate transcription factor activity with different functional states. And in particular, we observed that this cluster 10 cells, even when split between two different samples, acute and chronic infection, they are most similar to each other in this space, in sort of this reduced space of transcription factor activities, as they are in the genome-wide, with respect to genome-wide profiles. And encouraged with these results, we then also looked at actually a more granular, more fine-grained analysis of the single-cell RNA-seq data that we generated in the same model. And we also identify, well, we use this data also. We built this big atlas of CD8 T cell states that are possible at early and late time points, and actually even before the infection. And we use this also to look at, again, at the loci encoding transcription factors. And this way, we also prioritize transcription factors associated with different functional states. But we also identify this cluster, which we believe is progenitor dysfunctional cells. And we show that it is present, again, not only in the chronic infection, but also in acute infection, both at day 7 and at day 14, particularly day 7 after an infection, at the peak of the response to acute infection. And in this very small subset of cells, actually, some very well-known markers that are now associated with this progenitor dysfunctional cells are also observed as being overexpressed. So now this leaves us with the situation where our main observation is that from both single-cell ataxic data and from single-cell RNA-seq data, we have this striking similarity of the cells, which we call progenitor dysfunctional cells in the chronic infection, and their counterpart cells in the normal functional response to acute infection. these cells are very epigenetically and transcriptionally similar to each other. After all, that's why they form the same cluster in both single-cell RNA-seq and single-cell ATAC. So the question then is, how plastic are these progenitor cells? Do these progenitor cells retain the potential to differentiate along the effector cell trajectory? And in order to address this question, we performed an adoptive cell transfer experiment where we sorted the progenitor cells from the established chronic infection, and then we adoptively transferred them into the new host. And these mice were infected then with the LCLV Armstrong, which typically results in acute infection. And then at day 7, after expansion, these cells were profiled with single-cell RNA-seq. And we analyzed this data together with the previous samples, and I think this is actually what made this analysis quite powerful. We were able to have a reference of all potential, we think, CD8 T cell states, and now we can judge about this new state, what it is. And it turns out that despite the fact that the majority of cells in these mice are undergoing a normal response to acute infection, this small subset of donor cells, donor and expanded cells, actually are most similar to what we observe at chronic infection at day 7. So these progenitor cells somehow extracted from the chronic infection somehow retain their dysfunctional fate even under the acute viral challenge. So if we want to understand where this commitment to functional dysfunction happens, we need to look even earlier than day 7. Earlier, closer to the time when each individual T cell was actually activated, which might also be quite asynchronous. And I think it's a challenge in the next, it's a big challenge for us and many others. And with that, I want to thank my amazing collaborators, and most of this work was done when I was a postdoc in Christina Leslie's lab at the Memorial Sloan Kettering Cancer Center, which is an amazing place to do science. And I'm very thankful to funding, including some of my own, that I managed to secure for that project. And I refer you for many more details about this study to a publication that recently appeared in a Molecular Cell. And I also want to say that now I have my own lab, which is a computational, primarily computational, but also partly experimental lab. And we are very open to collaborations, and we're very happy now to connect with more people, particularly in Russia. And we are actually even hiring at multiple different levels. Now I think I'm very happy to take questions in English or Russian.